Our next speaker really needs no introduction. She hails from the University of Iowa as a distinguished professor of pediatrics. Her research and clinical practice focuses on pharmacologic and molecular characterization of G protein, the epidemiology of NETs in children, and theranostics for NETs. She has been an NIH-funded investigator since 1976 and mentors graduate and medical students as well as MD and PhD fellows in NETs. Dr. O'Dorisio has published over 150 peer-reviewed articles and has provided research support for additional publications involving various topics related to neuroendocrine tumors in children. And I'd like to say there's no one else like her in the world.
Thank you, Lowell, and thank you all for staying on the late after Saturday afternoon to hear what I have to tell you about genetics. And really, I'm going to tell you that you've learned an awful lot of genetics already before I get up to here to speak, because pretty much everyone who's spoken to you has talked about genetics in one way or another. What I'd like to do with you today is go back to the basics, actually, and review for you what we usually learn in college and what my grandkids are now learning in high school about chromosomes and genes and proteins. Then I want to demonstrate how we look at genes and proteins in neuroendocrine tumors and what ones are specific to neuroendocrine tumors, and then discuss a little bit about how we target them for diagnosis and therapy of neuroendocrine tumors. Now, if you look at the cell, we've talked a lot about tumor cells, and I look at the nucleus of the cell here as being the bank for our genetic code. And then inside the bank, there's another little area that I call the safe in the bank, and that's where these chromosomes are stored. If you take apart a chromosome, then you can see the DNA, but even then, the DNA is still protected by some kinds of proteins that cover it up and don't let it get damaged. If you unravel that, you can see a sequence that we call a gene. And then when we are actually sequencing your DNA, we actually look at every single one of these bases in that gene. So you can see that it is a pretty complicated system and that our body works hard to try to keep us from injuring that DNA. Now, I'm going to talk a lot about genes by looking at the protein that they code for. And how does that happen? Well, from the last slide, you saw the double helix and the nucleotides here that code for the gene. That has to get transcribed into an RNA. And we can now sequence also RNAs, but it has to be pretty complicated. And if Dr. Howe in our group at Iowa is the one who actually is the expert in this. And then you come down to the proteins, and they get transcribed. The RNA gets transcribed into proteins. So what are genes? You can look at them from several different angles. You can look at them from the DNA angle, and from the cell angle, you can say, well, I've got about 40,000 genes in every cell, but they don't all get transcribed. And you could look at them, as we look at them a lot, by looking at the proteins that are produced from those genes. And we look at those, and you can see right off the bat, as I listened the last day and a half, the two proteins from genes, or the two genes that I've heard the most about, are Ki67 and the somatostatin receptors. So I'm going to show you those two genes and how we look at them. We heard about them in both Dr. Halfdonis's very first talk on Friday morning, and we saw his pictorial depictions of the somatostatin receptor gene in that talk as well. And then our pathologist talked more about both of these genes. That was Dr. Laura Tang. So look at the nuclear staining. What I want to point out to you is they're going to look the same because we always stain them with a brown stain. But if you look closely under the microscope, you can see that some of those are going to be located in the nucleus, like the Ki67 always is, and some of them are going to be membrane proteins. Now, it's a little harder to see, but if you look carefully at this membrane staining, you can see that some of these cells, you can see the outline of the cell by a dark brown color, and that's actually staining of the somatostatin receptor that's in the membrane. Okay, let's go back to the chromosomes that I told you are stored in the nucleus of the cell, and ask the question, do we ever use those in identifying neuroendocrine tumors or trying to find out and classify neuroendocrine tumors? And you can tell we have a molecular pathologist who works at Iowa very closely with our team, and Dr. Darbrow is a cytogeneticist. He can take cells, live cells, and grow them in the laboratory and then look at their nuclei, and inside the nuclei he can find all the chromosomes. So I want you to notice that the chromosomes are numbered according to their size, starting with the largest one and going all the way down to this small one. And notice that sometimes you can see by this method whether someone has an extra chromosome. That could, I'm showing you here one that we're all familiar with, and that as a pediatrician I'm familiar with, and that's the way we diagnose trisomy 21 still in this day and age. But then there are the sex chromosomes, 2 X's and 1 X, or 1 X and 1 Y. So does one chromosome make a difference? Well, X chromosome, some of us in this room have 2, and some of us have 1 X and 1 Y. So right off the bat, one chromosome doesn't make a big difference, right? And if you look at these chromosomes then, it's actually known that the X chromosome has a tumor suppressor gene, and that tumor suppressor gene has been shown to be important in neuroendocrine tumors. If you were to lose one of those chromosomes, if I lost one of them, I'd still have one tumor suppressor left on the other X chromosome. But if a man loses one, they lose that tumor suppressor completely. Now, the testis-specific gene is not a tumor suppressor gene, but is one important sex-determining gene that is only found on the Y chromosome. Now, my son-in-law tells me that he's discovered another gene on the X chromosome. He calls the slip, trip, and fall gene, and he said, I got 2 copies of it and probably more, and I gave all of them to my daughter. On the other hand, I noticed when I was raising 2 teenage boys that I think they have a toilet seat gene. And my husband still has the other gene, I'm sure, is on that one. It's the channel changing gene. Now, how many genes are on each of these chromosomes? Remember I told you that we number them according to their size. The smallest one is the Y chromosome, and it only has about 200 genes on it. The X chromosome has about 2,000, but remember that number 1 gene has over 5,000 genes. So what chromosomes are related to neuroendocrine tumors? Well, you can see, I want you to look at the lung, the losses of 11q. So Q means the small tip of a gene, of a chromosome, and you can see losses of 11q in both lung and well-differentiated pancreatic tumors. Gains of 9q would actually be can be found on these same pancreatic tumors, and they would be, those are the kinds of changes you can see if you do a cytogenetic analysis of some of the fresh tumor cells. Now, small bowel tumors are characterized by a loss of 18, and that's a pretty big loss. Can be sometimes the loss of that whole chromosome. And then you see, as I talked about before, you can have losses of that tumor suppressor gene that's located right in this area. And I'm pointing out here that can be loss either of that DNA, or it can be turning off that DNA by methylating the area. Now, chromosome losses are pretty important in neuroendocrine tumors. That 18q that I just pointed out to you has been demonstrated, loss of that has been demonstrated in large studies to be actually from 50 to 80% of all small bowel tumors. The 9p again is an increase in a little piece of that DNA, and that's in about 50% of tumors. 11q is where the MEN gene is. So you can imagine that that's very important in pancreatic neuroendocrine tumors that are associated with the MEN1 syndrome. Again, I point out to you that tumor suppressor gene that's on the X chromosome, and that tumor suppressor loss is found in quite a few tumors, not just a single one. And let me explain what I mean by a tumor suppressor. That's a gene that slows down, usually it's a gene that slows down the proliferation of a cell. And then 3p beta-catenin is one that has not been investigated very much, but could be very important because, as you see, it can be in almost 50% of the intestinal neuroendocrine tumors. Now, probably almost all of you have heard about the 2-hit hypothesis, that for most genes, one exception being the X chromosome in a male, we would have 2 copies of every gene. Sort of an insurance policy that if one of those goes bad, you still can have the function of that gene in your body. The Nobel Prize was actually given for this discovery. Was hypothesized quite a few years, maybe 20 years before it was actually proven. But you can see that if you have a tumor suppressor on one arm of the X chromosome, for instance, and you have another one on the other X chromosome, you could lose that one and still have one that might function. But when you lose 2, you get a cancer. So what is the effect of having this, what I call a double hit? We call a hit, often we call it a mutation in the scientific language, and it can increase activity, it can decrease gene activity. In some cases, you completely knock out the function of that gene. I made a notation not of every single gene that was mentioned here in the last 2.5 days, but certainly I think probably most of you have heard of these, and I classify them again in peptide genes. We've talked a lot about somatostatin and gastrin, and we just heard about serotonin, and we've talked a lot about chromogranin and where to get it measured. Those all come proteins from genes. And then we've talked about a lot of receptors. VEGF and EGF receptor are usually treated by anti-angiogenic factors when you have those overexpressed in a tumor. And then inside the cell, we talk about cell cycle modulators. I think Dr. Lowell gave you a wonderful explanation this morning of how the cell cycle works. And we're finding now that Ki67, mTOR, which everolimus targets, and MEN1, which we measure by gene sequencing usually, but don't really know how it functions yet, are all genes that are important in neuroendocrine tumors. We all want to know what genes we inherited. The red proto-oncogene, these are all ones that are important in various forms of neuroendocrine tumors. Paraganglioma is a neuroendocrine tumor, and pheochromocytoma is as well. So you can see the VHL gene and the NF1 gene are important in both pancreatic tumors and in pheochromocytoma and paraganglioma. What we mean by this is not that they are going to, if you have a gene mutation in VHL or NF1, you're going to get a neuroendocrine tumor, but the incidence is actually increased in those families. And finally, the BRCA gene, which my family has, and we didn't find out until, I had a grandmother and several aunts. My mother, and several aunts die from breast cancer or ovarian cancer. And now, recently, there has been more information coming out. I actually did a search on this to see how many neuroendocrine tumors I could find where this was a genetic finding, either in the tumor itself or in as an inheritable trait. And it's been seen now. I found papers that reported it in pancreatic neuroendocrine tumors, in neuroendocrine tumors of the antrum, the small bowel, and the lung. But there's not been, it's a low incidence, I think, and that's the reason we don't have more studies on it. So more genes that take a hit that we know about that take a hit in neuroendocrine tumors. You see up there at the top, MEN1 and BRCA have been found in lung tumors, as I just showed you. In gastric tumors, VMAT is actually the transporter that takes radiolabeled MIBG into cells. So that's one that's important as a target for therapy. Small bowel nets, CDKN1B is a protein that was identified in a series of neuroendocrine tumors by Matt Kokia and his colleagues at Dana-Farber Institute, and then Dr. Howe followed up on that in his series of genetic studies of small bowel tumors, and I'll show you a little bit about that in a minute. So you can see that in every kind of neuroendocrine tumor there are some, but I can't find one that is in all of them. And that's part of the issue I think we have with neuroendocrine tumors. You can't find just one gene and say, hey, this is going to help you diagnose every single neuroendocrine tumor. It just doesn't happen. So let's look a little more closely at the CDKN1B and see, this is a cell cycle gene, but how would we study it in neuroendocrine tumors? Well, this is some work from Dr. Howe and Dr. Bellizzi, our pathologist, and they are looking, I'm going to show you across the top from their work, a primary tumor, there we go. This is a primary tumor and a liver metastasis and a lymph node metastasis from the same patient. Now at the bottom here, you can see there is CDK1, which was stained with an antibody, and that's why it's showing up brown, in some normal tissue that was surrounding a tumor that they resected. But up in here, in the area of the tumor itself, you don't see any CDKN1B. And here in the metastatic piece from the liver, you can see a little bit of brown up here, but in the tumor itself, there's no CDKN1B showing. And similarly, in the lymph node, it's very difficult to find. So in this particular tumor, they did find a mutation, and this particular mutation caused a loss of the gene itself, the DNA, so in fact then you lose the function of the gene as well. Now down on the bottom, these are tumors, and here you see normal tissue. You see how much normal tissue can express the gene, and there is some CDK1 there, but it just doesn't look quite right. And so if you look at it under higher power, you can see that in this area, I think, is one of the easiest to see. This is a clump of cells, and see the holes in them? Those are the nuclei. Well, this gene is supposed to function in the nuclei, and it got synthesized, but it wasn't able to be carried into the nucleus, and so it didn't function very well. And this is an example of how, even when you're looking at genes, just sequencing the gene isn't enough. You have to look at what its function is going to be, and our pathologists help us a lot in that arena. Now, we often use one single gene to look at something very specific in a tumor. So when and the first way that we usually look at that, if we can, is with immunohistochemistry. That is, having the pathologist take an antibody to that gene, that protein, not to the DNA, but to the protein that gene makes. And you've seen today that we have looked at. We can do that with CDKN1B, somatostatin receptor, Ki67, and I haven't shown you, but it's often in every pathological diagnosis, one of the mainstays that the pathologist does is a chromogranin A. So these are all very important in pathology, in terms of your actual diagnosis, as well as in further characterizing your tumor. Now, we can measure genes in the blood, and the ones we measure in the blood are chromogranin A and many of the peptides that pancreatic tumors make, or in the case of small bowel tumors, we often measure gastrin or serotonin. It's also not the gene, the DNA that we target when we try to make a diagnosis. We target the somatostatin receptor itself, the protein, when we use a gallium dotatate or dotatate PET scan. And in the other area of theranostics in which this is currently used, and was actually the very first use of theranostics, was with 123 MIBG for imaging and 131 MIBG for therapy. And in both cases, the imaging and the therapy, the target is actually this transporter that takes the MIBG into the cell. And finally, we're going to talk a little bit about when do you need to actually sequence some genes. So I think the question you all really want to know about is, when should I have genetic testing? And I'm going to go back to the American College of Genetics and the recommendations they give. So if you have a first-degree relative, a parent, a sibling, or a child, and you would think child wouldn't need to be in here, but I can tell you there are multiple cases in which I have seen a child found the gene, and then we go back to test the parent to find out where that gene came from and whether it is hereditary, and whether it's hereditary or whether it's what we call a new mutation. Now, if you have, so having a first-degree relative is probably the number one reason to have a gene sequenced or to have genetic testing. And by genetic testing, I don't just mean getting your gene sequenced, but I also mean getting genetic counseling. If you have one of the genes that actually aren't primarily a cause of neuroendocrine tumors, and here I've listed 3 genes in this case, but they're associated with an increased incidence of neuroendocrine tumors, that may or may not be a reason for you to get genetic testing. And then, of course, if you want to do it, a few years ago would cost anybody could do it, but would cost about $100,000. Today, it costs about $10,000 to do it if you want your whole genome sequenced. And no insurance companies pay for that I know of yet. So these are the official American Society of Clinical Oncology guidelines. TP53, BRCA, MEN1, and SDHB, which is a pheochromocytoma gene, are all reasons that are classified as number 1 because insurance companies would be, I can't say required, but more than 90% of insurance companies would pay for your genetic testing under these conditions. It's a little more difficult to get genetic testing for VHL and NF1, but many larger centers and hospitals would have a clinic. For instance, at the University of Iowa, we have both a VHL clinic and a neurofibromatosis clinic. And then you have some help to determine whether you need genetic counseling and whether you need any sequencing done. And then finally, syndromes, but no evidence of any gene mutation are very difficult. So for carcinoid syndrome itself, with no tumor identified, would be very difficult to get that genetic testing. So that you understand when a physician says to you, yes, I can help you get genetic testing, it really relates to how difficult would it be for us to convince your insurance company to be able to get this testing done. Now, this is a very interesting study about risk that came out of Sweden. And first of all, I'm going to talk to you about risk along the y-axis there. And the risk was studied. In Europe, they have a lot more global registries than we do here in the United States. And they can go back to their records and look at questions like this. So the question was, if a person in your family, a first-degree relative again, a parent, a sibling, or a child, has cancer, what is your risk of getting cancer? And you can see that it's not much increased. If you have diabetes, now isn't this interesting that the risk for you is a little higher than even if you have a person with diabetes than if you have a person in your family with cancer. And by diabetes, this meant type 1 diabetes. It didn't mean, it did not include type 2 diabetes. But then, if you have a first-degree relative who has cancer and diabetes, your risk went up to about 50-fold. Now, I wanted to put this in context for you because if you go back to the fact that 6, let's say 7 people out of every 100,000 have a neuroendocrine tumor, then 50 times that is still not a very big risk, right? When you get down to it, it's less than a 5% risk, more like 1 to 2% risk. So you can sometimes, these graphs can sometimes be misleading, but they do make the point that, you know, you can get an idea from this about what you should be thinking about in terms of your own health. And this one's the other way around. I often get this question from parents who have a neuroendocrine tumor. What's the risk of my children having that? And you can see it's about 10-fold increased. But again, put that in the context. Instead of 6 people or 7 people having it for every 100,000, this would only be up to 60 out of 100,000. So again, it's not a high risk, but it's significantly more than if you did not have it. So what are the genetic tests that are available to us as physicians and to you as patients? So I want to just run through the ones that are most common. That's certainly not all of them. But I think I tried to give you some that are in each category. One, immunohistochemistry. We have at the University of Iowa a pathologist who is very interested and committed to neuroendocrine tumors and has developed tests for a lot of the antibodies. Almost every hospital is going to have an antibody for Ki-67. Our pathologist has also develop stains for the somatostatin receptor itself and for CXCR4, which is a target that is being found in many high-grade lung carcinoma, both bronchial carcinomas and bronchial neuroendocrine tumors. That test requires something that almost all of you would have. If you had a biopsy to determine your diagnosis, then and there's any tissue left from that initial biopsy, you would have the material you needed to have an immunohistochemistry test performed for these or any other genes if you had a good antibody to them. Cytogenetics, on the other hand, examines the chromosomes in live cells. So in this case, you have to have the surgeon send live cells down to the cytogenetics lab, and they have to grow those in the lab and then be able to sort out the chromosomes, as I showed you in that very first slide, to be able to look at every chromosome individually and see if you have 2 of everything. Is there anything missing from the 18? Is there any extra for the 9 chromosome hanging around? That takes live cells. Foundation One is a private company that actually uses the paraffin-embedded tissue or blood, and by using the paraffin-embedded tissue from your tumor as well as your blood, they can look for genes in the tumor that only have the tumor mutation, but they can also, from the blood, then examine it at the same time and say, is this in all your other cells too? Meaning, is this hereditary mutation that you have? Now, gene sequencing can be done. It's most economical and most readily performed if there's a gene, such as the VHL gene or the NF1 gene or the MEN1 gene, that has already been found in a first-degree relative. In that case, they can sequence, they already know the mutation they're looking for, and they can sequence only that gene. It's much faster, and it's much cheaper to do. Someone asked a question about the Ren test. I'll just tell you what I know about the Ren test. You can do it on blood, and they use 51 genes, but it's not public knowledge, I think, what those 51 genes are. They include some genes that are important in neuroendocrine tumors and some genes that they have mathematically determined may be important in some of the pathways that are important in the proliferation of these tumors. I think the question of how applicable it's going to be is going to be partially determined by the same group of investigators who are developing what is called the OncoTest. It's probably maybe 4 or 5 years away from being developed, because with that test, they're going to try to take your genes, and they're going to take your cells, and they're going to grow them in the laboratory and then be able to test them against a group of drugs, maybe up to 500 different drugs, maybe up to 1,000. They did a little preliminary test on this in neuroendocrine tumors and came up with a protein kinase inhibitor that hadn't heretofore been used, and I think they're beginning a clinical trial to test that gene. So that one's a way off. So what do I think is important for you to know about genetics when you have a neuroendocrine tumor, or why would you want to know about it? I think you can understand your own disease better if you understand that there maybe have been caused by mutations, even if you don't know what those mutations were. I think it helps you converse better with your doctors, and especially if you have under consideration whether or not you need to have genetic testing, that you understand what you're asking when you ask for genetic testing. Anytime, as I stated before, that you have genetic testing done, you will probably be referred to a genetics department, and they will actually give you genetic counseling if you are found to have a mutation. You know, some of us don't want to know what all our genes are, don't want to know what mutations we might have, and if you get whole genome sequencing, you may find out ones that you wish you hadn't found out about, and that's that's actually become an ethical question about who should receive genetic testing, and that came up mostly with Huntington's disease. If you diagnose that in a 5-year-old, but you don't, and you know that by 33 they're going to have the disease, but there's no way of fixing it, how ethical is it to do that kind of testing? So there are important questions that do require genetic counseling. If you want to request coverage, or you want your doctor to request coverage from your insurance company to have genetic testing, I think it's important for you to actually know what and for your doctor to know. So sometimes your doctor is going to need to have a geneticist advise them on whether it's indicated or not. I do that quite often. You know, as I said, I've had several patients who were diagnosed with a cancer as a child, and then how do you cancel the parents of that child? So I hope you have a little better understanding of where chromosomes and genes are in the cell, and particularly in a tumor cell, what the chromosomes, how they store the DNA, and this actually is the scientific definition of a gene, but there's a whole lot more to the story. So I think it's important to know that chromosome losses and gene mutations can cause cancers, including neuroendocrine tumors, although it's a very muddy question, and we're just beginning to understand the genetic causes of neuroendocrine tumors. And I heard one of our speakers this morning say it's a very personal interaction between you and your doctor, and the genetic makeup of your tumor is definitely one of those areas that's a very personal, personalized to you, and we can't yet determine that for any single neuroendocrine tumor. We can, however, exploit these mutations or gene expressions that we know, and we are exploiting them for diagnosis. In the case of Ki-67, for diagnosis and therapy and follow-up of the response to therapy in the case of somatostatin receptors, and thinking about imaging those tumors. And I believe that the future treatments will target multiple genes, because it takes you can imagine if you have 40,000 genes in every cell, it takes a real interaction between those to guide how a tumor cell is going to divide and multiply. So we have a ways to go in that area. want to thank you for listening, and we will have more, and I'll be happy to answer questions when we have the question and answer series.